nos acompañan desde otros lugares del mundo. Mi nombre es Ana Silva, como encargada del Laboratorio de Neurociencias de esta facultad. No se imaginan la alegría y el honor que es estar dándoles la bienvenida a esta primera edición de la conferencia anual Ruben Budelli. Esta conferencia la organiza el Laboratorio de Neurociencias como actividad destacada de las jornadas de la Sociedad de Neurociencias del Uruguay y con el auspicio de la Federación de Sociedades de Neurociencias Latinoamericanas y del Caribe, FALAN. Como verán, esta conferencia se va a dictar en formato híbrido, ¿verdad? Y antes, entonces, de dar comienzo a la misma, quería agradecerles realmente a todos y todas la presencia hoy acá, a nuestros participantes presenciales, a nuestros participantes virtuales, a la gran conferencista que aceptó la invitación y muy especialmente a los familiares y amigos de Ruben que se acercaron hoy acá y que están en la audiencia, tanto acá con nosotros como a través de la pantalla del Zoom. Y para dar comienzo formal, la profesora decana Mónica Marín va a darnos un saludo de bienvenida.
Bueno, muy buenos días a todas, a todos. Es un gusto enorme tener este pedacito de tiempo para darles bienvenida a esta conferencia, que en primer lugar quiero agradecer, reconocer la iniciativa, que realmente es sumamente importante para nuestra institución, tener esta conferencia anual Ruben Budelli, por todo lo que significó Ruben para nuestra institución, como científico, como docente, como compañero de trabajo tan cercano con los demás docentes, con los estudiantes, con todo el personal de la facultad. Ruben marcó, digamos, un estilo, una forma de vivir la ciencia integrado en la comunidad de la facultad. Así que más que contenta con esta iniciativa, reconociendo, digamos, esta permanencia de Ruben a través de estas conferencias que sin duda van a ser todas de excelente nivel y nos van a marcar, inspirar para nuevos trabajos, nuevas ideas, nuevos proyectos. Muchísimas gracias por estar acá y un éxito en estas jornadas.
Muchas gracias, Mónica. Bien, a continuación le voy a dar la palabra al doctor Daniel Herrera. Daniel Herrera es asistente del Laboratorio de Neurociencias y en este momento está desarrollando un postdoc en UPenn, Pensilvania, Estados Unidos, y fue de hecho quien tuvo la idea, la iniciativa de realizar esta conferencia anual que hoy se materializa. Adelante, Daniel.
Hola, veo que está un poco desactualizada la foto, pero bueno. Bueno, gracias, bienvenidos a la primera conferencia anual Ruben Budelli. Primero, un par de puntos operativos. La charla va a estar en inglés, pero quienes están viendo desde el Zoom pueden agregar subtítulos en las opciones. Después se pueden hacer preguntas con el botón de Q&A en Zoom, en el chat de YouTube y levantando la mano en el salón de actos. Y el chat de Zoom está abierto para saludar, pero no se pueden activar ni las cámaras ni los micrófonos. Bueno, y ahora paso a introducir un poco la conferencia Ruben Budelli, y para eso voy a hablar de los dos grandes objetivos que motivaron el organizar la conferencia. El primer objetivo de la conferencia es ser una instancia para traer investigación de punta de otro lado del mundo a Uruguay, en lugar de tener que irse el investigador uruguayo para afuera. Pero pedirle a investigadores de alto nivel mundial que nos presten el tiempo y el esfuerzo que implica prepararnos una charla tiene sus dificultades. Cuanto más uno avanza en reconocimiento, mayor es la lista de responsabilidades, de invitaciones y de mails no respondidos. Entonces, al invitar a una persona exitosa a nivel internacional a que se detengan a explicarnos su ciencia, es importante darles un buen motivo para hacerlo. De esa necesidad surgió la idea de organizar lo que en inglés se denomina una named lecture. en Neurociencias en Uruguay. Estas son charlas donde se homenajea a una persona importante en el área, que suelen tener un cierto esfuerzo organizativo detrás y que se hacen de forma anual o cada un par de años. De esta forma, reservamos como comunidad un espacio que acordamos es importante, tanto por el homenaje realizado como por la atención que nuestra comunidad científica le presta a la charla. Esto hace que nuestra invitación a dar la charla sea también un reconocimiento de nuestra comunidad hacia la persona invitada y por lo tanto un atractivo para que compartan su tiempo con nosotros. Así también generamos valor en nuestra comunidad, resaltando la relevancia de las personas que homenajeamos y reconociendo la importancia de nuestra atención en un escenario internacional. Entonces, esta conferencia es también un reconocimiento a la importancia de nuestra comunidad científica. El segundo objetivo de la conferencia es homenajear al profesor Ruben Budelli. De la idea de hacer una named lecture en neurociencias, Ruben surge de forma inmediata como la persona homenajeada. Ruben fue un pilar fundamental del desarrollo de las neurociencias en Uruguay. Formado como físico, fue un pionero en aplicar las ciencias computacionales a los sistemas biológicos en el país. Desempeñó cargos en Facultad de Medicina y Facultad de Ciencias y ayudó a tender puentes entre diversas áreas científicas en nuestra comunidad. Fue una persona muy comprometida con el desarrollo de las instituciones científicas de Uruguay y también formó a varias generaciones de neurocientíficos en el país, con lo que su influencia en nuestra comunidad neurocientífica actual es enorme. Entre otros, Ruben fue mentor de Leonel Gómez, mi tutor de doctorado, y por lo tanto es mi abuelo académico. Pude conocer a Ruben en los últimos años de su vida y por eso no me extraña el cariño y admiración con que se acuerdan de él sus estudiantes y colaboradores. Ya con unos cuantos años de carrera científica, puedo ver que Ruben tenía unas cualidades como científico y profesor que lo hacían muy especial. Cuando uno comienza la carrera científica, ve la ciencia como una expresión de la curiosidad y creatividad, un deseo por aprender y por divertirse descubriendo los misterios de la naturaleza. Es una aproximación a la ciencia con una mentalidad de deslumbramiento infantil en el mejor sentido de la palabra. Al avanzar en la vida académica, la realidad después va cambiando y nos encontramos que nuestra mentalidad al hacer ciencia muchas veces se aleja de ese juego infantil. La necesidad de publicar papers y acumular puntos académicos, entre otras, son muchas veces nuestra motivación más inmediata. Ruben era una persona que parecía siempre tener intacta esa chispa de curiosidad y de disfrute, esa aproximación jovial a la labor científica. No solo disfrutaba de aprender, sino de compartir sus conocimientos que transmitía con profundo entusiasmo. Aunque como profesor de pelo gris y enorme físico tuviera un aspecto intimidante para los estudiantes jóvenes, al hablar con él uno era tratado como igual y se podían tener divertidas y profundas charlas sobre neurociencia, matemática, evolución o sobre la vida. En esas charlas Ruben mostraba siempre una enorme humildad, desinterés y generosidad de su tiempo y conocimiento. Todas estas son cualidades que trascienden cualquier métrica científica, que creo que todas las personas que conocimos a Ruben admiramos y que han tenido su efecto en cómo se hace ciencia en Uruguay. Por ello, nos alegra que nos acompañen en celebrar la memoria de Ruben y esperamos que esta conferencia ayude a mantener vivo el ejemplo de todas esas virtudes que quienes lo conocimos admiramos. Y ahora le paso la palabra a Ana para que presente a nuestra invitada a presentar la charla.
Gracias, Daniel. Bueno, es un verdadero honor para mí presentar a la profesora Carmen Sandi como disertante de la primera edición de la conferencia Ruben Budelli. Carmen es directora del Laboratorio de Genética del Comportamiento del Brain Mind Institute de la Escuela Politécnica Federal de Lausana en Suiza. Comenzó sus estudios en su país natal, que es España. Realizó su doctorado en el Instituto Cajal y la Universidad Autónoma de Madrid y continuó su formación en 2 estadías postdoctorales en el INSERM de Burdeos, Francia, y en The Open University del Reino Unido, respectivamente. Poco después, Carmen se establece en Suiza y desde hace 20 años es profesora en la Escuela Politécnica Federal de Lausana, donde ha consolidado su línea de investigación en la búsqueda de los mecanismos por los cuales el estrés y la motivación y la personalidad y las diferencias individuales afectan la función cerebral, el comportamiento, con especial énfasis en la motivación y el ámbito social. Su enfoque es abarcativo y le han permitido combinar estudios en modelos animales, en humanos, y a través de abordajes que incluyen comportamiento, cognición, bases moleculares, metabólicas e imagenológicas. Ha publicado alrededor de 200 artículos, lo que ha dado como resultado más de 12.500 citas y un índice H de 64 para quienes estamos al tanto de lo que son esas cosas. Carmen Sandi fue directora del Mind Brain Institute, presidenta de la Sociedad Europea del Cerebro y la Conducta, presidenta de la Federación de Sociedades Europeas, codirectora de la Red Suiza de Investigación sobre Trastornos Psiquiátricos SYNAPSI y más recientemente fundadora y primera coordinadora del ALBA Network, organización que ha revolucionado el abordaje de los temas de inclusión y diversidad en la neurociencia global. No conozco personalmente a Carmen, casi no puedo creer decir esto, porque he interactuado y trabajado con ella en forma bastante activa en los últimos años. De este conocimiento, no dudo en decir que Carmen es la mujer científica activa que más admiro. Admiro su ciencia, pero también admiro su forma de hacer ciencia. Creo que Carmen ha transitado un camino que nos muestra cómo establecer liderazgos de maneras diferentes a los que nos precedieron en otras generaciones. Liderazgos que se basan en el entramado de redes, en liderazgos que se basan en relaciones horizontales, liderazgos que hacen crecer a las personas alrededor y que marcan políticas científicas que yo entiendo son las políticas científicas de la neurociencia del futuro. Así que se imaginan entonces que este es un día muy especial para el Laboratorio de Neurociencias, muy importante. Tener a Carmen Sandi hablando aquí para nosotros es un privilegio sin precedentes y sin duda el mejor homenaje que podríamos hacerle a nuestro querido Ruben. Así que muy bienvenida, Carmen. The floor is yours y muchas gracias nuevamente por aceptar esta invitación.
Thank you so much, Anna. It's a great pleasure and honor to be giving the first conference for Ruben Budelli. It's a name that it's new for me. I didn't hear many times this family name, but I read now about Ruben Budelli's trajectory and also the interdisciplinarity that he tried to put together, and also the spirit of indeed being inclusive and bringing neuroscience and work at the highest excellent levels. And I'm very proud and honored that you asked me to give this first lecture on his name. And also I thank you a lot for the introduction. It was quite emotive for me. So I'm going to try to upload my presentation, and then you will tell me if it's okay or not, because one never can give for granted that's going to work. Let's hope. Not yet coming for me.
I see the slides.
You see it? Okay, thank you. So thanks again, and I am also very proud and happy to share with you the work that we've been doing in my laboratory. I think what I'm going to present here started exactly 10 years ago. It started with a bit of intuition, and I think a lot of interest in understanding behavior from a neuroscientific point of view. This image, I think, acknowledges the importance of understanding the brain in order to understand social behaviors. I think it's very clear what's going on here. There are two males that are trying to establish a social hierarchy, or at least that they are trying to decide which one is going to have access to resources. But of course, the work in my lab is not with rhinos, or it's not in the wild. It's not a zoological work. We do a lot of work with rodents and also in humans, as Anna indicated. And we've basically tried to have models in which we can have social competition, also social hierarchies, and also sociability as the focus of research branch from the laboratory. So recently somebody told me that they were very surprised to hear in the same sentence about social competition and mitochondria. And I think that deserves a bit of introduction. Our work goes maybe one step beyond from a lot of work that it's being performed at the moment in the field of neuroscience, in which what we are trying is to understand the circuits that explain particular behaviors. So there is perhaps the tendency to understand the brain a bit as if it's cables that connect and that are involved in specific functions. And I think this is very important work, and it's very important to understand which connections and which projections and which downstream mechanisms are involved in particular behaviors. But I have the feeling that sometimes we should be careful that we don't forget that the brain is not only cables, that inside the cells there are many organelles, not only molecules, because there is been a lot of molecular neuroscience work, but also there are organelles, and many of these organelles have a life on its own. And I think what is coming up in the literature at the moment, it's showing that there is also computation or a contribution of organelles that is very important for how neurons and other brain cells function. And that's maybe the framework of the message that I want to bring today. A bit of more introduction, because we started focusing on mitochondria, and there is the tendency also to think that mitochondria is mainly an organelle to produce ATP, the powerhouse of the cell, as it's frequently termed. But it goes, the functions that mitochondria has goes well beyond only producing ATP. It's very important for many metabolic processes, and also it's very important for the production of reactive oxygen species, and also for also many other functions, like, for example, apoptosis, and also even autophagy, which is coming up also as a very important process in neuroscience. Something else that I think is worth mentioning is that mitochondria is at the core of the production and also degradation of many neurotransmitters. So if we can imagine, because I'm going to be talking about differences in mitochondrial function, and there are many functions, but among the functions of mitochondria, it's the production of the main neurotransmitters. Glutamate and GABA are produced in mitochondria, starting from precursors like glutamine, for example, and then glutamate, and in GABAergic neurons also GABA. And also many of the neurotransmitters are also metabolized, like, for example, dopamine in mitochondria by enzymes like MAOA, for example. And something that's important when we talk about behaviors, like social behaviors, but also stress, which we do a lot of work, and you will see that my talk is going to revert also on stress and also anxiety, because we try to understand the individual, and these are very powerful functions. Then the main stress hormones, but also other hormones like glucocorticoids and also sexual hormones, are also produced in the mitochondria. So if we think about it, mitochondria can be considered like messengers of mitochondria or mitokines. Cortisol, for example, or glucocorticoids can be considered mitokines, not only hormones as frequently termed. And another very important point of introduction for my talk is the following, because in indeed, I presented the images in which mitochondria are organelles inside cells, inside neurons, and one could maybe imagine that they are there only at the expense of the activity of the cell, as slaves of synapses to produce energy whenever it's needed. But in fact, they have the capacity to be regulated by many other components. And one very important one is that indeed, going back to these hormones, cortisol, for example, is produced in the adrenal glands, then it circulates in the bloodstream. And we know very well that it's capable of crossing the blood-brain barrier and crossing membranes and affecting many processes at the level of gene transcription, including the transcription of many proteins that are important mitochondrial proteins, and also the activation of glucocorticoid receptors by glucocorticoids can also lead to the translocation of those receptors into the mitochondria, where they can also affect transcription for mitochondria DNA, but also other functions in mitochondria. So what we have here is the idea that we can have changes in brain mitochondria that are not due to the activity of the brain circuits, but are due to changes in periphery, changes related to a stress activation and also other metabolic processes. So I think with this introduction, we can understand maybe in a different way results like the ones we started getting when we applied chronic stress in mice. And we have here the results of transcriptomic analysis, in which what we did was to give mice 28 days, 4 weeks of chronic stress, and then analyzing the transcriptomic changes in different brain regions. In particular, I'm showing here for the prefrontal cortex and the nucleus accumbens. And what we observed is that, particularly in the prefrontal cortex, there were many mitochondria-related genes that were differentially expressed or affected by the chronic stress. And then when we analyzed other data sets in the literature from other chronic stress models in mice and also from depressive individuals, what we observe is a very similar enrichment, very similar particularly as we have here on the right side for the increase in expression of mitochondria DNA genes. So we did behavior in those animals to look for different depressive-like behaviors. Like, for example, we looked at sociability, and there was a reduction in sociability due to the chronic stress. And what we can see here is that when we represent the social avoidance index, we see a huge variability in those animals that are chronically stressed. It happened the same for other motivated behaviors. So we did indexes, and then we decided to look at the data according to how much the individuals had been affected, whether they were vulnerable, highly vulnerable in the changes in behavior, or more resilient. And when we represented the expression of mitochondria DNA genes according to this stratification in the groups, we could see that those that were low in effect, if you want, low in susceptibility to stress, they looked very similar in the expression of these mitochondria DNA genes for different complexes to the controls, very similar. However, those that were more affected, the high susceptible individuals, they show huge differences, huge changes in the mitochondria DNA. So this brings me to, I think, one of the main topics of my lab and interest, which is the existence of individual differences. Yes, chronic stress leads to depression that involves low motivation, also for social activities. We see here that it's represented normally as individuals that are alone, either actively seeking for social avoidance or maybe as a consequence of the behavior, but many times depression goes with these alterations as a result of stress. And also anergia is a very important component of these conditions, and chronic stress leads to depletion in the energy sources of the organism. So this is one way to maybe depict the message that I want to bring. Stress, indeed, particularly when it's chronic, can affect individuals in different ways. Some of them are more resilient, others are more vulnerable. And we've been trying in the field to understand which are the mechanisms that differ between one and the other, particularly at the level of brain differences in different brain types, brain regions and cell types, etc. But what we were particularly interested was in asking if this vulnerability could come from a priori conditions, if we could identify traits that a priori allow us to identify individuals that are highly vulnerable to stress. And this brought us to do a lot of work, a lot of behavioral work with batteries of tests of spontaneous behavior and application of principal component analysis. And to make a long story short, what we found is that one trait, which is a personality trait, both in animals, that's what we show in the lab, but afterwards we've done a lot of work and then verified that this can be applied also to humans. The personality trait of high trait anxiety is one that allows us to identify individuals a priori. The way that we see it is that trait anxiety is a kind of a continuum, like following a Gaussian distribution. And those individuals that are above the mean of this Gaussian distribution are more vulnerable when they are exposed to stress to develop alterations, particularly at the behavioral level, also, of course, at the neurobiological level. And those individuals that are at the extreme of high anxiety, they already show quite important comorbidity with depression. And here, because my focus today is on social behaviors, I have to say that this is true when we talk about motivated behaviors that require effort in general, and it's true also for social behaviors, as we are going to see in the next minutes or so. So at the moment, we use the trait anxiety and the test that allow us to classify individuals according to this trait as a way to identify individuals a priori, to identify different vulnerability to stress. But I have to say that this goes beyond only stress. It's a vulnerability because maybe also because of the stress, which brings a lot of perturbations to the physiology of the organism. It's a trait that eventually is also a vulnerability trait for other conditions, particularly also neurodegeneration, as we are seeing that's coming importantly in the literature. So what we've been doing in the lab that brought us to these discoveries I'm going to share with you today was to take rats. That's how we started with rats. Then we continued with humans. And then more recently, we do a lot of work in mice as well. We characterize them for anxiety according to their behavior in a battery of spontaneous behaviors for exploration. This is the heat map of the behavior of low anxious individuals in the elevated plasmase. I think you might be maybe familiar with this test, but basically in all the cases, all the tests, the open field with the novel object recognition and the LiDAR box, what we give the animals is a trade-off between conditions in which they can feel safe and other conditions that they have to choose what they do, whether exploring the areas where they are safe, and that means that avoiding the open spaces, avoiding the center of the open field, and avoiding the lightened spaces, or to explore more, which I think it brings also many advantages from a kind of a more adaptive process. And what we see is that we have huge dissociation between the animals that we are testing. We also have animals that are intermediate. I will show you some of the data of those. But to make it clearer, what we show here is the huge differences between those that are classified as low anxious versus the ones that are high anxious that really avoid completely these anxiogenic areas. And when they are classified according to this and follow an index of anxiety, then when we test them in other behaviors that probe for motivation in general, like anhedonia test, we see a tendency in this case for high anxious animals to drink less sucrose solution. Of course, something important here is that this test doesn't require any effort, so it's probably less sensitive to these differences. But then in the force-swing test, which is a test of coping with adversity and having maybe an idea of whether they can cope with their behavior by floating or versus swimming, then we see a huge difference, and high anxious animals show much higher immobility. In terms of social behavior, the social competition test or social dominance requires a lot of also energy. And when we put two animals, two males of equal characteristics, equal body weight, equal conditions in the laboratory from beginning to end, except for the trait anxiety of the animals, we see that high anxious animals have a disadvantage to win a social competition. It's not that they are not motivated, they really try, but eventually with time, they don't manage. It might be a kind of a computation of not being able, even if they have probably the same characteristics, because we know very well that if we give them an anxiolytic, then they even can win the social competition. So this way of looking at the individuals and identifying different vulnerability allow us now to classify animals before any fighting or any stress or whatever, just understanding which ones are high anxious and low anxious, and ask if we can identify neurobiological differences that could perhaps already explain a lot of the vulnerability to stress and also to differences in the coping in social behaviors. So originally we did experiments in which we put animals to compete and looked with tests for brain activity and asked which brain regions were showing differential activation due to this experience. And what we found among the top ones, top brain regions that were showing differences, is that the nucleus accumbens was massively activated from the baseline, the comparison. Other brain regions that were also activated was particularly the medial prefrontal cortex, which is in well agreement with the literature, but the nucleus accumbens had a superior increase. And that's very interesting because indeed the nucleus accumbens is a brain region that is important for the exertion of effort, also for reward learning. And it's also been found in both animal and human studies that it's activated when the individuals are given situations like incentive, motivation, effort, and following chronic stress, and also in depressed individuals, the nucleus accumbens shows a very important alteration and also a causal alteration in the behaviors that are induced. Something I think also interesting is that the nucleus accumbens, of course, it's a hub in the motivated brain, but it's also coming as a very important brain region affected by the anxious circuits. So, okay, we knew that we wanted to look at the nucleus accumbens, but there are many different cell types in the nucleus accumbens, and we needed to know in which cell types we needed to look. So we did a co-staining with immunohistochemistry and looked at different cell types. I'm only going to show you here a few of the results, but we did several experiments. And in the nucleus accumbens, similarly to other parts of the striatum, also in the dorsal striatum, what we have is a very important representation of medium spiny neurons that constitute around 95% of the neurons in the nucleus accumbens. And then we have other neurons, like, for example, cholinergic interneurons, GABAergic interneurons, etc. So we looked for markers of the different neuronal types and co-labeled with CFOs in order to ask if we could identify which cell. In the median spiny neurons, there is a very important distinction also in the literature, which is between those neurons that contain the dopamine receptor 1 or the dopamine receptor 2. There is also co-expression of these receptors in a few subsets of neurons. And the way we did it was by co-labeling with some of the peptides that are co-expressed with these neurons. And what we found was that the D1 containing or expressing median spiny neurons were particularly activated, and they were particularly activated in the low anxious animals. It's as if during this social competition, high anxious animals didn't really manage, we could say that in that way, to activate these neurons. Of course, we needed to know if these neurons and also the nucleus accumbens activity is important for the outcome of the social competition. And what we did was to do both transient inactivation by injecting mucimol, small amounts in the nucleus accumbens, and this was injected in one of the two animals that were paired in that case for equal anxiety. And we could see that this was showing a disadvantage for the animal that was injected versus the vehicle one. And also we injected an antagonist of the D1 receptors, and then the animal that was given this treatment also show disadvantage to win the social competition. So then what we asked next is if we characterize the animals and we stratify them for anxiety, would it be that there are differences in the median spiny neurons? We started looking at the morphological level, and what is depicted here is the Golgi analysis of low anxious and high anxious animals for the median spiny neurons. And here is the quantification with a Scholl analysis of the number of intersections at different radius that give us an idea of the complexity of this arborization. And as you can see, it's very different. High anxious animals have a poorer dendritic complexity. They also have less spine density. And then in particular, what they have is a lower density of the mushroom or mature spines, which is the important site for the connectivity and synapses. Then we repeated these experiments because we were doing electrophysiology, and then we labeled the cells with biocytin and confirmed that this difference is there for the median spiny neurons and also in particular for the D1 containing median spiny neurons. So this reassures us that we were in the maybe good direction. In the electrophysiological experiments, we further confirmed that there was the high anxious rats display a lower frequency of excitatory inputs onto these median spiny neurons, indicating poorer connectivity. So this reflected and supported the findings at the morphological level. Then, in order to maybe advance a bit more, we wanted to know if we could identify important molecular pathways that could explain this phenotype. And we ran at that time microarrays. And when we performed the gene ontology analysis, we found an enrichment of mitochondria and metabolism genes that were differentially expressed between high and low anxious animals, really pointing at the importance perhaps of these organelles and pathways in differing the two type of phenotypes. So we started asking whether mitochondrial functions might be different. So what I show here is results obtained with Ouroboros. It's an ex vivo analysis of the maximal respiratory capacity of the mitochondria when they are given substrates in order to see each complex, how far it can go in the consumption of oxygen. And as you can see, this is for complex 1, both complex 1 and 2. We have a deficit from the very entry into the mitochondria electron transport chain, that there is a deficit in the functioning. We also found a deficit at the level of protein level. High anxious animals really have alterations in mitochondria. And at that time, we thought, because of the results that we had, that it was particularly in the neuronal function, not in glia. Now, I don't have time to show you also because it's ongoing work, but with electron microscopy, we also see differences in glia mitochondria that we are exploring at the moment. In the nucleus accumbens, then we also measure ATP, and high anxious animals have lower levels of ATP, and they have higher levels of ROS, or reactive oxygen species. So this was pointing us really towards mitochondria as a possible important mechanism, but correlation is not causality. So in order to ask if this was relevant for the phenotypes that we were observing, what we did was first plotting the maximal respiratory capacity for the different complexes versus the level of social dominance. And we already saw a very interesting linear correlation and positive correlation that was pointing towards the importance there. But then we did interventive studies by injecting inhibitors of either complex 1 like rotenone or complex 2 for both competitive and non-competitive inhibitors, 3-MP and malonic acid. And as you can see, those animals that were injected with these tiny amounts that we verified that they were not inducing toxicity in the brain, just mimicking the differences between high anxious and low anxious. I have to say, we did these experiments by taking now the intermediate anxious animals, and they were matched for equal anxiety, so that one of them was injected with these inhibitors and the other with vehicle. So we put them to compete, and we could see that those that had this treatment, they had a disadvantage in winning the social competition. And then we also discarded the other alterations in behavior. So we did a lot of controls. And I think what it was also interesting is that we could do the reverse experiment in which we gave NAM, which is nicotinamide mononucleotide. This is a form of precursor of vitamin B3 that boosts respiration. It boosts the mitochondrial function, and this also boosted the social dominance. So when we injected it to the high anxious animals, they didn't have any more the disadvantage in competing against the low anxious animals. So we started looking at mitochondria images to see if we could gather more important insights. Originally, what we did was to count the number of mitochondria, also mitochondria DNA, with PCR, and we at that level of mitochondria density, we didn't see differences. So we thought that perhaps indeed there should be something else that was different. As you can see here, what we did was a 3D reconstruction of a cube of tissue. And then, yeah, looking in the second step, we had the first paper already published. There was a second postdoc that came to the lab and who was really expert in morphological analysis. He said, look, I see huge differences in the shape of the mitochondria. And then when we did this 3D reconstruction, it was very obvious that low anxious animals have mitochondria that are like thinner, and they don't occupy massively all the space of the neural tissue, whereas the high anxious animals have a more swollen mitochondria that you can see also here. It's the mitochondria is depicted in cyan color versus the smaller mitochondria. So mitochondria in the high anxious animals covers a higher area, also more coverage of the area. And then we quantified the also contacts with the endoplastic reticulum, because this kind of association, these interactions of these two organelles are very important for calcium buffering, also for processing of lipids. And voila, we found a huge difference, showing that high anxious animals have much less of these contacts than what we see in the low anxious animals. This made us think that perhaps molecules that are important for the mitochondrial dynamics, for fusion, fission, and also for the interaction with endoplastic reticulum might be affected. So we ran a pipeline of this test to test for the different molecules. This is now RNA, mRNA data that I'm showing here for different molecules. We have the first one is mitofusin 2, mitofusin 1, DRP1, OPA1. And then eventually we ran also protein level with Western blot, with immunohistochemistry, etc. What we systematically found is that this molecule, mitofusin 2, is the one that it's always really differently expressed, much lower expressed, lower levels in the high anxious animals, as you can see here also for the protein level. And we found it that is less expressed in both D1 and D2 expressing median and spiny neurons. And because I'm talking so much about the nucleus accumbens, we looked also at other brain regions like the VTA and also the amygdala, and there we didn't see differences. So what is this mitofusin 2? Mitofusin 2, perhaps as the name indicates, it's implicated in the fusion of mitochondria. Mitochondria are very dynamic organelles, and these fusion and fission processes are critical for many functions. So it's very important to have the molecules on place. So we already see that we have a deficit at this level, and this is reflected eventually in this phenotype that I was indicating. Then it's also implicated in motility, and this makes us believe and think that perhaps this is also an important function because we have less dendritic complexity and also less spine density, and it's going to be very important to have mitochondria located very close to the spines, and if motility is affected, this could be another mechanism. Then this is a very key process and function of mitofusin 2, is this tethering with endoplastic reticulum, and that we already observe we have alterations at that level. So the critical experiment for us, and the next one, was to say we have low anxious and high anxious animals having lower mitofusin 2. In order to really test for causality here, what we did was to develop a virus, an adeno-associated virus, in order to re-express mitofusin 2 in the neurons of the nucleus accumbens, because we were not working with mice at that time. We couldn't be cell-specific for the median spiny neurons, but we were specific for neurons. And then we did this now by having a high anxious a group that was injected with this virus, another group that was injected with a sham virus, and also controls. And then we look at the different phenotypes that I just described for you. And in all the cases, everything that we tested, the re-expression that is expressed here in yellow of mitofusin 2 in the nucleus accumbens of high anxious animals made the measurements that we undertook look as if we were measuring low anxious animals. We reverted the phenotype of high anxiety to low anxiety. Here is for the mitofusin 2 levels, but also for the morphology of mitochondria, as you can see, also for the function of mitochondria, for oxygen respiration. For the dendritic morphology, we have the poor arborization in high anxious that it's reverted in the overexpressed ones. Now at the electrophysiology level, what we find is that these neurons are incorporated in the circuitry, and then at the level of behavior, it was quite remarkable to see that not only social dominance and the coping behaviors were reverted, but also even anxiety was reverted. So what I show you until now, it's going from behavior to maybe identifying nucleus accumbens cells as differently expressed, or different phenotypes, different morphology, different function. And then we could go and find that mitochondria was also different in many components, and then identify this mitofusin 2 that is causally implicated in this all these phenotypes. So as I told you, we could now perhaps say that mitofusin 2 was important in nucleus accumbens neurons, but we couldn't conclude from the previous work that it was only specific manipulation of the median spiny neurons, and then also whether we could have a different contribution of mitofusin 2 and also mitochondria in the D1 versus D2 type of neurons. So what we undertook as the next step was to now take mice in which we have flux mice for mitofusin 2. And then what we did was to inject viruses in the nucleus accumbens that were targeting either the D1 median spiny neurons or the D2 median spiny neurons. The data coming particularly from the D2 median spiny neurons, we published it very recently in Cell Reports by Gosal. I'm going to give you maybe the highlights of this work. We didn't publish yet the D1, it's ongoing work, but what we already find that it's very interesting is that indeed there is a segregation of the functional behavioral consequences of manipulating mitofusin 2, downregulating mitofusin 2, either in D1 or the D2 median spiny neurons. In the D1, we see that it's more anxiety and also more the coping behaviors, motivated behaviors. And in the D2 ones, particularly, in this case it's for mice, maybe it's a bit confusing because when I was talking about rats, I told you that the D1 were very important there. At least we saw main differences. But here now, when we specifically target the different subtypes in mice, it's very clear, as you will see soon, that the downregulation of mitofusin 2 in the D2 expressing median spiny neurons affect social behaviors. So the first evidence or the first question we wanted to see is if we mimic now with this approach. Now we don't have a complex phenotype like in the rats, that maybe many things were different. They are also outbred. They are genetically heterogeneous. Now in mice, we have inbred animals. Everything is the same. They've been considered for many years as biological replicates. And what we do is specifically now to affect one projection, one cell type in one specific brain region. And then we see that we see many of the phenotypes that are very similar to the ones that were expressed in rats that were highly anxious. So, for example, when we look at the maximal respiratory capacity of mitochondria, we see that this leads to a reduction. Also, the ATP to ADP levels, they get reduced. Also, the interactions with the endoplastic reticulum in a cell-specific manner, in this particular neurons with ligation SA, we see that they are reduced. Now, at the level of the morphology of the neurons, the dendritic arborization gets also affected, is less complex, and then they also have a poorer integration into the circuitry. So this fits very well in this model that mimics very well what we see in the rats. And then when we look at the sociability, this is mice that have been cohabitating in the same home cages. It's not just a acute social competition. We test the social hierarchy with the tube test and also with the worm spot test. And then what you can see very, very clearly is that the winning is much lower in those that have a downregulation of mitofusin 2. So this is something that we see across days and also in the worm spot test for social competition. So I conclude from this part that we identified mitofusin 2 as a very critical protein, mitochondrial protein. Is reducing high anxiety, modulates mitochondrial, neuronal structure and function, regulates motivation and social competitiveness, and the behavioral and neurobiological impact is different when it's downregulated in the median spiny neurons D1 or D2. I have to say mitofusin 2 is upcoming as a very important also hub for disease, if you want, or psychopathology. Maybe we can discuss this during the questions. So something that we've been very interested in asking is what's the origin of these differences? Because if we go back now to the rats, they are genetically heterogeneous. What could be the difference? Of course, we didn't do genetics, and I think it might be very complex to do genetics because many genes eventually interact, are polygenic, etc. But we wanted to see if also there could be differences in the functioning of the mesolimbic system. The mesolimbic system is constituted by the projections from the ventral tegmental area to the nucleus accumbens. This is a very important dopaminergic projection, and dopamine We have other publication that I don't have time to indicate here. Is a huge modulator of mitochondrial function in the nucleus accumbens acutely. So that's acutely. Once dopamine transients are gone, then the trait comes back. But we wanted to see if there were differences at this level, and indeed, we have several papers in which we describe different physiological and also molecular components that are different in this system. So this is something that might be contributing. Something that we are exploring at the moment, and we are very excited, but I don't have time to tell you, it's also ongoing work, is the possibility that the high anxious animals might have a more compromised blood-brain barrier at the level of the nucleus accumbens. We started thinking about this because work by the colleagues, Menard and also Scott Russo, working with chronic stress, they found that nucleus accumbens seem to have in their hands a particular vulnerability for this leakage or compromise of the blood-brain barrier. And we look in our high anxious versus low anxious animals, and we see that this seems to be the case, even though we don't apply any stress. So we are trying to understand whether this could be another component that could contribute. Something else that, of course, one can ask, and particularly because we are so interested in stress, is whether perhaps because early life stress makes individuals more anxious and also more prone to develop depressive behaviors in adulthood, could it be possible that early life stress could be programming this system? So we took advantage that we were doing a project for something else in the lab, and then we could ask this question with the same data. What we did here is to apply a model of peripubertal stress that meant to expose mice. These were now all equal, so we only want to see the effect of stress and not of maybe other types of variability. We gave stress during puberty, and then we tested the animals when they were adult, and we verified that they are more anxious. They also were less sociable. And then 2, 3, even 4 months after stress at puberty, we asked at adulthood if the nucleus accumbens would have differences in the maximal respiratory capacity of the mitochondria. And as you can see here, that's what we observed for the complex one, again, being highly affected. And then we looked at the electrophysiological properties of the median spiny neurons, and we observed that they had a lower firing frequency. We also found that they had less levels of NAD and other components that are very relevant for the functioning of the mitochondria. But then, because in that study we wanted to look for many other components, we included as well in a transcriptomic, spatial transcriptomic analysis, genes that were relevant for the mitochondrial dynamics. And of course, we included mitofusin 2. And then when we look at this contrast, this is now 3-4 months after the peripubertal stress. At adulthood, we see that mitofusin 2 is reduced, and is reduced in the D2 median spiny neurons specifically. There were also reductions in the D1, but they didn't reach statistical significance. I don't have time to tell you about that study, but I think it's important for the message of this talk that not everything comes from the brain. What we observed here is that early life stress or peripubertal stress changed also the body composition, the body fat composition, and the adipocytes were atrophied. And this led us to investigate a lot this lead, whether there would be factors from fat that could be changed in a kind of a long-term manner that could maybe affect the composition of blood adipokines, and whether this could be signaled in the brain. And that was what we found. We could describe a kind of a fat to brain or to the nucleus accumbens pathway in which the fat was affected by the peripubertal stress, and in particular to produce lower levels of NAMPT, which is secreted to blood, to the extracellular form, and this could eventually affect the levels of NAD and also the excitability of the median spiny neurons and sociability in the brain. I I'm aware that I have maybe 2 or 3 more minutes. So I've been talking so much about nucleus accumbens. Is it so relevant, or are we biased? So I can tell you that now we have another project also in the hippocampus. We find that high anxious animals also have alterations in mitochondria in the hippocampus, and particularly also of the mitofuscin 2. So this is something that perhaps could also contribute, but we are looking at this at the moment. And I will finish giving you the highlights of a recent paper published by a number of people, particularly by the group of Martin Picard, also contributed by our lab a little bit. And this paper, what was asking is indeed, can we take maybe a more global approach and then see if we can explain behavior from what we learn about mitochondria in different brain regions? And the protocol was the following. The idea was to say, in humans, we have a lot of variability that is due to different types of experiences. So instead of maybe looking only at one experience, why don't we increase variability by exposing animals to one group to chronic stress, chronic social defeat, another group just giving them chronic corticosterone, another group is control animals not submitted to anything. But what they did was to test them in a number of anxiety-related tests, elevated plasmase, open field, novelty suppressed feeding. They took then samples from the whole brain. As you can see here, these are punches of 17 brain regions, and also samples from other organs, and then performed enzymatic activity and assays for tiny volumes of tissue, and then afterwards probe for gene expression, particularly for genes of the respiratory chain, and also different functions, and providing afterwards an index of mitochondria health. And what this study shows first is that when we look at the activity of mitochondria and features related to mitochondrial health in many different brain regions and across the body, it's mainly the common variance is mainly shared across the brain. So the brain mitochondria seems to be more co-existing, co-functioning than with the rest of the body, which I think is very reassuring when we are thinking about behavior. And then what they try to do is to see if they could identify shared variance across the 17 brain regions in a similar way to what is done in functional MRI, functional magnetic resonance imaging. And indeed, there were like what it's called here networks, 3 networks of brain regions in which mitochondria share common variants, and they can be like clustered. So the first one is a corticostriatal one, the second one called salience, spatial navigation, and the third one threat response. And then when looking which of these networks related to the behavior in different tests, open field, it's mainly the network one that it's correlating with the anxiety behaviors. And from this, I think I better pass it here because it's bigger. So it's mainly this one, the first one. And from these different brain regions that are included in network one, the only one that survives multiple comparisons for significance is the nucleus accumbens. So it seems that, again, going from a global perspective, nucleus accumbens comes up as a very important one. So why doing all this? I think we have a lot of intellectual curiosity. It's a lot of fun, but this also has been inspiring treatments, and we've done quite a lot here. And now we are very excited with a lot of new approaches. We've published several of them in the literature across the years. We've tried and found successful results when probing or providing supplements with nicotinamide riboside, with NMN. This was in the context of the sociability that I just talked to you, urolithin, that it's boosting mitophagy, a very important process, medium chain triglycerides that are providing ketone bodies and that are very important also as a kind of help to mitochondria in a way of producing less ROS, L-acetylcarnitine, taurine, etc. I don't have the time, but perhaps if somebody's interested, we can discuss later. So I finish here. Still, I think there is a lot to do. We are now very reassured that mitochondria metabolism are critical for neural coding and also for defining motivated behaviors because of the brain regions that we are targeting, and also social behaviors, and that identifying anxiety and stratifying for that helps us a lot defining the individual. So I would like to acknowledge many collaborators over the years, and also the ones that are indicated from previous former lab members, current lab members, etc. And I include here in particular the ones that have contributed to this project. The upper row is former lab members, the second one current lab members, our wonderful lab technicians, and the human group. I didn't have time to tell you about it, but we also do a magnetic resonance spectroscopy in order to look for metabolism and also to look at how this can be related to behavior. And I would like to thank you for your attention. This is the campus of the EPFL, and this is the Lake Leman, and this is the pre-alps in there. Thank you.
Thank you very much, very, very much. Excellent conference. We might have some time for questions. And of course, las preguntas se pueden hacer en español o en inglés. Y si alguien tiene alguna pregunta, también se pueden hacer en forma virtual o a través de la plataforma. Tenemos una pregunta acá. A ver si la puedes escuchar, Carmen.
Sí.
Hello, thank you. Should I do it in English or Spanish?
Como quiera.
Por favor.
My name is Patricia Casina from Facultad de Medicina, Montevideo, Uruguay. Fantastic talk. May I speak Spanish or?
Sure, yes. Sí, sí.
No, porque el idioma nativo siempre es más fácil. Preciosa charla, me pareció muy estimulante y muy impresionante. Me gusta ver esto del metabolismo, shaping, el fenotipo celular, que es algo que hago una cosa bien interesante, y quería hacerte pregunta a propósito de lo que tú comentaste, que la disfunción mitocondrial o la disminución en la función mitocondrial era específica en las neuronas espinosas y no en la glía. No sé cómo hiciste esa medida, si separaste las células por cell sorting y luego Ouroboros, o simplemente por la evaluación morfológica. La pregunta es porque me importa mucho evaluar la función por la morfología, que es como una tendencia actual, que el solo ver la morfología mitocondrial nos hace predecir cómo está su función respiratoria. Esa primera pregunta en primer lugar. Y después, si evaluaron, si bien la glía no tiene alteraciones en la función mitocondrial, como tú dijiste, si evaluaron la captación de glucosa o la liberación de lactato, porque como bien sabemos hay un acoplamiento metabólico entre estos dos tipos celulares, y la función mitocondrial en la neurona depende del aporte de lactato a veces de la neurona. Quizás bueno, tú dijiste que ibas a buscar alteraciones en la barrera vascular, pero bueno,
sí.
Dejo por ahí.
Muchas gracias por la pregunta, es muy estimulante. Al principio, cuando empezamos a trabajar en todo esto, casi no había nadie, por decir nadie, trabajando, y fue bastante difícil. Entonces, simplemente conseguir hacer Ouroboros en aquella época, que esto fue en el año 2014, en un volumen tan pequeño como el núcleo accumbens, nos llevó casi 2 años, porque intentamos establecer colaboraciones y la gente creía que no iba a haber diferencias, que necesitábamos un knockout o algo así. Entonces, bueno, fuimos ahí persistentes Y al principio vimos estas diferencias y sí, claro, queríamos intentar tener en lo máximo posible especificidad celular. Pero al no trabajar con ratones era complicado. Sí, hicimos fax, intentamos, pero mitocondria se queda fatal, muere casi, y no podíamos hacer después estudios funcionales. Entonces lo que hicimos fue unos sinaptoneurosomas en donde vimos que teníamos las diferencias que habíamos observado en la máxima respiración y la parte que no eran los sinaptoneurosomas, que contenía todo el resto, incluida la glía, ahí no había diferencias y por eso pensamos en aquel momento que era sobre todo neuronal. Y al ser median spiny neurons el 95%, pues eso apoyaba mucho que quizás estaba ahí la diferencia. Después sí vino un postdoc y trabajamos en lo que yo he comentado ahora. Después tuve también otro postdoc que se continuó en el laboratorio, que venía con experiencia en astrocitos y le propuse que mirara a los astrocitos y empezamos a ver diferencias muy claras también de los contactos en mitocondria con el retículo endoplásmico en astrocitos, pero no en todos. Los astrocitos que están en torno a la sinapsis no los vemos tan afectados. Los que vemos muy afectados son, es que sé las palabras en inglés, los que tienen el end fit que cubre la unidad neurovascular, los que están en la parte del endotelio. Esa parte de astrocitos, por una parte, cubren menos en los animales altos ansiosos, tiene menos cobertura del astrocito, del end fit del astrocito, y también las mitocondrias ahí están más pequeñas y tienen menos contactos con el ER. Ahora estamos haciendo el experimento en el que estamos, también tienen menos mitofusinados y hemos desarrollado ahora un virus para hacer target, para afectar solamente los astrocitos, la expresión de mitofusinados y ver si cambiamos la morfología celular, la conducta, etcétera. También, como tenemos alteraciones en la barrera hematocefálica, claro, se pueden explicar por muchos componentes, pero uno de ellos puede ser por estas alteraciones en los astrocitos. Estos astrocitos que estoy comentando son los que expresan aquaporina 4, que es muy importante para el paso de nutrientes desde la circulación al cerebro, como saben, y están también reducidos los niveles de aquaporina 4 en los animales altos ansiosos. Entonces, ahora mismo estamos haciendo todos esos experimentos, hemos hecho medidas metabólicas. Ahí no hemos visto cambios en lactato, pero no lo hemos hecho después de un fax y de un cultivo. También porque el problema con los cultivos, que es algo que yo tenía mucho interés en hacer, es que la predicción mayor con todas las personas que estoy hablando es que se pierde el fenotipo. Y eso es por lo que tenemos ahí un reto importante para ver cómo podemos hacer las medidas, depende de cuáles sean, por supuesto. Entonces, sí, a nivel metabólico, en metabolomics hay diferencias, pero no de lactato. Eso no quiere decir que si lo estimuláramos hay diferencias, pero eso es lo que puedo decir ahora a nivel de astrocitos. Sí creo que algunos, al menos una parte, están afectados. Y también hemos hecho single cell single cell RNA transcriptomics, sequencing. Así que podemos ver la expresión de genes en todos los tipos celulares y ahí sí tenemos muy alteradas las células endoteliales y los astrocitos también. Y lo que es muy interesante es que al hacer estudios de interacciones entre distintos tipos celulares, la que nos está saliendo muy diferente es las interacciones entre las células endoteliales y los astrocitos. Es una posibilidad que yo pienso es que quizá hay también un factor periférico que está afectando la barrera hematoencefálica y que de alguna forma los astrocitos pueden estar afectando también el transporte de precursores también a las neuronas. Pero ahora mismo estamos haciendo todos esos experimentos.
Muchas gracias.
Gracias.
Daniel, ¿tenés preguntas virtuales?
Sí, hay una pregunta acá de Cecilia Escorza. Gracias, Carmen, excelente la charla y muy motivadora. Nuestro foco en función mitocondrial está siendo relacionado con procesos neurodegenerativos e inflamatorios. Hay dos preguntas. ¿Has testeado parámetros de inflamación en animales con altos niveles de ansiedad? La segunda pregunta es más específica. ¿Es condición suficiente ver desde el punto de vista comportamental si un animal está estresado o es necesario medir corticoides en plasma? Muchas gracias.
Gracias. Todo depende de la pregunta. Empiezo con la segunda. Los corticoides, si suben, indica que el animal está estresado, pero es difícil solamente a partir de los glucocorticoides definir qué animal está más estresado. No son índices por sí solos. Hay otras hormonas, por ejemplo, progesterona, que quizá pueden reflejar más el nivel de estrés. Lo que pasa es que este campo de la neuroendocrinología y del estudio hormonal, yo creo que se ha quedado un poco estancado. Seguramente va a venir de nuevo en el futuro. Ahora mismo, ¿qué es lo que define estrés? Es algo que atrae mucho interés también en los humanos, porque hay mucho interés por los aparatos wearables, para saber cómo identificamos estrés, cómo lo medimos, no hay una única forma. Yo creo que es todavía un ámbito difícil, muy complejo y de muchísimo interés. A mí sí, me parece que es muy difícil contestar a esta persona, ¿cómo se llamaba? Cecilia, ¿no? Me parece.
Cecilia.
Sí, yo creo que lo mejor es tener muchas medidas, todas las que se puedan, y después ver si hay clústeres de individuos que tienen distintas manifestaciones de estrés.
La primera parte era si habían medido procesos
Inflamación.
neurodegenerativos e inflamatorios asociados
Sí.
a alta o baja ansiedad.
Hemos medido en sangre y hay algunas interleuquinas que sí tienen tendencia por lo menos a responder más fuerte, como interleuquina 6. En el cerebro es obvio que cuando mandemos el trabajo a publicar de la barrera hematoencefálica van a preguntarnos por microglía, y es algo que todavía no hemos mirado porque miramos muchas cosas. Entonces yo he dicho, hay algún momento en el que quizá otras partes se pueden hacer en colaboración o algo así. Pienso que es importante. Quiero ver cuánto podemos avanzar sin tener que implicar a microglía. Seguramente, no tengo duda que va a estar implicada. Sí,
Bueno, dice Cecilia, muchas gracias, quedamos a las órdenes.
gracias.
Estamos un poco atrasados de hora, pero me gustaría si hay comentario o pregunta en la audiencia presencial.
Yo tengo
Allá tenemos una pregunta. Vamos a tomar una pregunta de la audiencia presencial y una virtual. ¿Te parece bien, Daniel?
Dale, la virtual era mía igual, pero dale.
Igual, pero la tomaremos.
Buenas. Muchas gracias por la charla, muy completa, como muchos temas.
Presentate, por favor.
Soy Santiago Bosch, trabajo acá en la Facultad de Ciencias. Me llamó mucho la atención cuando dijiste lo de la estructura de las mitocondrias, que se veían una diferencia en tamaño. Me hizo recordar mucho, digamos, donde he escuchado que pasa eso también es si tenés animales en situaciones de ayuno, digamos, o de saciedad. Que se ve lo mismo y también tiene que ver con la fisión y con la fusión de las mitocondrias de una manera media dinámica. Supongo que en los experimentos estos que mostraste, los animales deben estar todos alimentados de manera igual, digamos, y correcta. Y supongo que entonces ocurre medio aleatoriamente cuáles van a ser, o digamos, en principio no sabían cuáles iban a tener ansiedad y cuáles no. ¿Tenés idea si, o ustedes o si algún otro grupo, si hay una relación entonces con la alimentación que tienen estos animales, por ejemplo, y bueno, eso, si esos cambios en la estructura de las mitocondrias si llevan a efectos similares o lo que sea?
No hemos hecho estudios para ver si, por ejemplo, de privación de comida a un nivel alto lleva distinta vulnerabilidad o nos permite evidenciar distintas características, pero sí hemos hecho estos estudios, tienen todas las condiciones de comida igual y también después de una privación de 6 horas para que todos estuvieran en las mismas características energéticas y hemos visto más o menos lo mismo, no una plasticidad distinta. Lo que es muy interesante, yo creo que queda por explorar en detalle, es que, por ejemplo, en ansiedad y el de estrés temprano, como mostré, y también otro tipo de estrés, vemos cambios en la composición entre lean-mass y fat-mass, ¿sí? Y ese cambio metabólico pienso que es muy importante porque contribuye a una distinta aportación de nutrientes también, de los nutrientes que se secretan de la grasa, pero también de los músculos y todo esto. Yo creo que es fascinante y va a venir muy fuerte en los próximos años. Una anécdota interesante es que cuando hacemos la reducción de mitofusina 2 en las neuronas de tipo D1, hemos hecho después estudios de la composición metabólica del organismo y aumenta la grasa y disminuye la parte de limas. Es decir, que hay algo por ahí, saber exactamente cuál es el componente y poder decir, podemos excluir que hay diferencias en locomoción o en cómo comen o el patrón de comida o algo así. Es algo a explorar y también difícil luego de captar para saber causalmente cuál es el elemento. Gracias. Es todo lo que puedo decir ahora.
Gracias. Y hago mi pregunta, Ana, o ahí capaz en el piso, no sé.
Tu pregunta, tomamos tu pregunta.
Muy bien, muy bien. Bueno, muchas gracias de nuevo. Como decía Santiago, muy completa la charla. Me había escrito algunas preguntas, pero que las fuiste contestando a lo largo de la charla. Y te hago una pregunta ahí que me quedó, es el tema de la oxidación, las mitocondrias, está muy asociado, como decía Cecilia, a la inflamación, también al envejecimiento, y es algo que se estudia bastante en Uruguay, y que también a veces hay como procesos de retroalimentación de esas cosas, ¿no? Como que oxidación lleva a degeneración, más oxidación. Entonces te quería preguntar si, como estas diferencias metabólicas interactúan con la edad del animal, si se ve quizás un envejecimiento cerebral o que a medida que pasa el tiempo, como que los de alta ansiedad van empeorando más, o si son procesos más estáticos.
Gracias, Daniel. Me interesa mucho tu pregunta, pero no la hemos podido abordar porque hay que hacer algo y cuando hemos encontrado lo que hemos encontrado, hemos intentado buscar mecanismos, etcétera. Pero sí, sobre todo ahora que tenemos mucho interés también por tratamientos y hay algún tratamiento que afecta a la mitocondria y después tiene efectos a corto plazo, al menos muy buenos. Pienso que será muy interesante y muy importante también dejar a los animales envejecer y ver cuáles son las consecuencias a largo plazo. Quizá en este contexto, en este marco, vamos a poder abordar tu pregunta, porque si no, es muy difícil para nosotros hacerlo porque es carísimo tener los animales en el estabulario tanto tiempo y luego hacerlos, ya sabes, el envejecimiento es difícil estudiar, pero sí, me parece muy importante. Gracias.
Gracias a ti.
Bien, no sé si me escuchan porque acá tuvimos un problema con la conexión. ¿Se escucha mi voz?
Sí.
Escuchamos.
Ok. Entonces, simplemente con esto estaríamos terminando, agradeciéndole una vez más a Carmen. Creo que no me ves, Carmen, pero te
No.
quería dar un saludo muy especial y un agradecimiento final y decirles a todos, todas los que estamos acá presentes, que habremos confirmado el privilegio que tuvimos de tener esta conferencia y en nombre del Laboratorio de Neurociencias les doy una vez más las gracias y los esperamos en la próxima edición el año que viene. Muchas gracias a todos.
Muchas gracias a todos. Un placer.
Gracias.